This will be the preparation of the culture media. First, we want to calculate the weighing powder, which percentage you want to add peptone, NaCl, and we want to calculate and weigh the measurements under the weighing balance and add sufficient number of distilled water, either 100 ml or 500 ml according to our research purpose. We want to add a distilled water for the makeup purpose, and this process needs to be sterilized in autoclave, where I have mentioned you the temperature of 121 degrees Celsius, and after that it should be fine tuning with the supplements, and pH is a necessary. pH is also important for the growth of microorganisms, and after autoclaving it should be placed in the lab and it should be poured in the petri plate, and the petri plate, whatever the technique you want to streak or spread, the technique has to be performed under the lab condition or biological safety conditions, and they should be marked and labeled and should be observed on the next day. So coming to the types of culture media based on the consistency. So as I said before, the consistency plays a major role in performing the plating technique and through liquid technique. So solid medium, semi-solid medium and liquid medium. So solid medium, it main purpose to isolate the bacteria as pure culture. So if we want a bacterial pure culture, we will go for solid media, and the agar we should add up to the concentration of 1.5 to 2% of concentration you want to add. So in that 1.5 to 2% of agar concentration, the medium will maintain the solid environment and functions. It mainly supports the bacterial growth as colonies via streaking method. So we'll have several streaking methods. So in that streaking method, the individual pure culture of bacteria can be isolated by using this solid media. And colony identification, for colony identification, it should be the baseline for the microorganisms to grow, and several examples like nutrient agar, McConkey agar, blood agar and chocolate agar. So in this agar, the agar is 1.5 to 2% of concentration will be added, and the streak technique, the microorganism can be identified. So this is the first one is the nutrient agar, and second one is the McConkey agar, blood agar, and third one is the chocolate agar. You could see the individual bacterial colonies that can be isolated from the streaking technique. So this is the streaking technique where you have used your inoculation loop, and the bacteria is taken on the inoculation loop in sterile technique, and the plate should be inverted all over the sides, and it should be kept in the incubator, and on the next day the bacterial colonies will be visible as like this individual colonies. So coming to the semi-solid media, so this media supports the bacterial motility. So semi-solid in the sense the bacteria is capable of moving by the use of flagella, so it supports the bacterial motility, and agar concentration is 0.5 or less. So we want to add agar up to 0.5%, and the consistency is semi-solid in the sense we all know about the consistency like jelly like consistency. Examples towards media, amaze media and mannitol motility media, and this appear as the thick line in the medium. So this form of appearance is thick line in the medium. So you should clearly seen in this picture, these are all the semi-solid medium. So you could see the steward line, uninoculated tube where there is no line, and in inoculated tube you could see the straight line appearance of the bacteria. So this is the semi-solid. And coming to the liquid media, so the liquid broth is mainly aims for the bacterial growth, not for colonies, only for the bacterial growth, where we observe the turbidity. So turbidity in the sense slight turbid-like changes in the tube, and it is useful for the fermentation studies and conditions. Moreover, it is similar as like solid media, like 37 degrees Celsius for 24 hours. So in liquid media, no agar can be added, because when we add agar, it is a solidifying agent. But for liquid agar, we will not going to add any percentage of agar in that, so that the medium remains in liquid. consistency. An example, nutrient broth. So I like to share thing for you. So every agar, nutrient agar, McConkey agar, chocolate agar, it is semi solid medium, and every broth indicates the liquid medium. So the examples of liquid is nutrient broth, tryptic broth, and methyl red VP broth, and it is mainly the growth indicator. The turbidity is observed at the end of the incubation, the growth is there. If the turbidity is not there, the bacterial growth is not there.